started. Our first talk is by Emily Kiker Snowman. And Emily, we'll leave it to you. Take it away.
Thanks so much. Yeah, I'm Emily Kiker Snowman. I'm a postdoc at Yale University now, but going to be talking today about some work that came out of my PhD at the University of New Hampshire. I'm going to be talking today about a paper that I co-led with Danica Lombardozzi that's out now in Global Change Biology. And this paper was born out of originally a USDA grant that brought together community land model modelers with empirical ecologists from across the US with the general goal of reducing decadal scale uncertainty in our system models by integrating more terrestrial ecology into our system models. And so this paper really represents the culmination of learning to do the work of putting ecology into Earth system models and all the things that we learned along the way about how to build effective bridges between empirical and modeling communities. So that's what I'm going to be talking about today. This project is really born out of the understanding that ecology really matters in the Earth system. So many biogeochemical fluxes of matter and energy are controlled by individual organisms on the land surface, and those organisms obey ecological principles. So things like where an organism is located on the land surface, how it behaves throughout its life, and how it interacts with other organisms. All of these things are controlled by ecology, and all of these things impact the way that organisms mediate biogeochemical fluxes. So we listened to a lot of really cool talks this morning about fates, and fates represents a really neat opportunity to explore the role of the ecology of above ground plant communities in mediating biogeochemical fluxes. But we also know that below ground communities are really important in mediating biogeochemical fluxes. So soil microbes, for example, control the decomposition of plant litter, and soil communities determine how much of the carbon that's fixed by plants gets stored as soil organic matter or released back to the atmosphere as CO2. So both above ground and below ground communities that are governed by ecology play a really important role in biogeochemical fluxes. Modelers and empiricists both know two things. One, that Earth system models are a really important tool for understanding and projecting the future of global change. And empiricists in particular also know that models generally and Earth system models specifically are really useful for scaling ecological insights beyond the scope of an individual data set. And modelers and empiricists also both know that ecology really matters in the Earth system, as we just discussed. And so it's reasonable then to conclude that Earth system models should strive to include more ecological principles. But actually doing this work is really tricky. It gets really difficult to facilitate conversations between modelers and empiricists. And I'm sure many of you have had this experience as modelers where an empiricist has come to you, your empiricist buddy comes and says, I have this really cool data set. Isn't there some way that you can use it for your model? Or we know this one thing really matters for our system and your model doesn't include it. Can't you just add it? And it can feel sometimes in doing this work like this cartoon shown here, where the empiricist is sort of shoving data into this mystery machine. And on the inside of the machine, the modeler is sort of picking and choosing between the data and cutting it out into what they need to inform model development. And it can feel like there's not great communication between these two sort of disciplinary groups sometimes. But in doing this work, my team and I, in putting together this paper, really wanted to move away from the paradigm of dividing these tasks involved along disciplinary boundaries and towards a paradigm where modelers and empiricists are working together at every stage to co-produce knowledge. So for example, in the early stages, when an empiricist is gathering data and developing ecological insight, the modeler can still be involved in saying, oh, can you please make sure to measure these things? Or can you please make sure to understand how your process interacts with these other processes? Because those things are in models already and we're going to need them down the line. Then in the middle stages, empiricists and ecologists can work together equally to translate between the language of data and empirical insight into the language of equations and biogeochemical fluxes that models use. And then finally, even in later stages of integrating models into Earth system modeling frameworks, empiricists can still be involved in making sure that the way that data are used and the way that the processes are integrated within the broader scheme of an Earth system model, that those things are faithful to the on-the-ground understanding of, the on-the-ground empirical insight about how a process works. And so to facilitate this kind of co-production of knowledge and the conversations necessary between empirical and modeling communities to make this happen, my team and I developed this flow chart with the intention of trying to provide a relatively jargon-free, straightforward understanding of how this process actually works, what the steps are that are actually involved in going from having some new empirical insight to actually working that into an Earth system model. And I'm going to talk about this figure in a fair amount of detail, so don't worry about trying to read every single thing on the slide. But importantly, we want to move away from this sort of illusion shown at the top that the way this process works is that you sort of shove data into a model and you get insights out the other side, and towards this understanding that there are many steps involved in this process, and every single one of those steps has unique data needs shown in the white boxes and develops specific insights shown in the tan boxes. So my team and I sort of divided out this process into 3 phases in sort of increasing effort and involvement with a Earth system modeling team. So the first phase shown in green is meant to be a series of simple questions that an empiricist alone or an empiricist with a modeling consultant can ask to ascertain whether or not a process is even ready for incorporation into a model. Then in the middle stage, is all about building simple models and exploring whether they explain observed patterns at increasing scales. And then finally, the last stage shown in blue is requires involvement with an Earth system modeling team, is all about integrating that process into an Earth system model and exploring how the Earth system model behaves with the new process. And so I'm going to go through this flow chart, each of these stages individually, and talk about specific examples of processes that are sort of at different phases within this chart. But I encourage you, as I talk about it, to be thinking about your own empiricist buddy who has a cool data set, and think about how you might use this framework for having a conversation with them about how they might use their data in your modeling work, or about what the next steps are that are necessary for moving an ecological insight towards eventual implementation in our system model. So the first example that I want to give is locus swarms. Locus swarms are kind of a cool example because in the places where they occur, they're massive ecosystem forces. They cause these huge defoliations with all of these, downstream ramifications for ecosystem function. But to my knowledge, and please forgive me if I'm wrong, but to my knowledge, no Earth system model incorporates any type of fauna, let alone locus swarms specifically. So let's say hypothetically that you had a locus swarm ecologist buddy come to you and say, I have this really cool locus swarm data set, and I want to understand what's happening in the ecosystem using your model. Well, here are some questions that you might use to facilitate a conversation about what's necessary to move from that initial conversation into eventual modeling exercises. The first question is, does the process behave consistently through space and time? So models operate in the language of patterns. And if you can't reduce a process down to a consistent set of relationships between variables, it's going to be really hard to model it down the line. So you might ask your locus swarm buddy, okay, we can think about modeling this, but first I need to know, how does locus swarm emergence relate to climate and vegetation factors? And what is the locus swarm impact on vegetation going to be? Can you quantify those things for me in a way that's consistent across space and time? The second question that you might ask is this process already included in our system model? And this question is really important because empiricists often don't know what's already included in our system models. So in the case of your locus swarm buddy, you might go to them and say, okay, let's think about this. Have you heard of fates? And maybe you would have a conversation with them about fates and how you might use that framework to think about impacts on vegetation from something like a locus swarm, because that's going to facilitate model development down the line. That's going to make your life easier. And the last question finally is this process likely to impact climate on scales of time and space that are relevant to Earth system models? this is a really important question because there are a lot of really cool ecological insights that cannot be directly tied to climate feedbacks. So to give another insect example, you might have a friend who's really interested in mosquito species distributions, and maybe they can demonstrate that mosquito species distributions are really important for structuring the food webs of certain communities. But if you can't demonstrate that mosquito species distributions have a direct impact on climate fluxes, then it doesn't really make sense to go through the effort of generating a model with the intention of working it into an Earth system model to represent that. So then sort of thinking about this next phase, I want to move to a different example that's near and dear to my heart, which is the mimics model. Now, I understand that the mimics model is in the process of being integrated into CTSM, so it's perhaps not a great example anymore of this phase of the process, but for a long time, it did spend time in this sort of eddy of testing a process by itself. So this
You're
phase—
about 11 minutes, Emily.
Okay, thank you. I'll speed it up. This phase is all about building a simple model and testing whether it explains observed patterns at small scales and eventually up to the global scale if you want to think about incorporating something into an Earth system model. So with mimics, that took the form of comparing against litter decomposition data sets, and now with global runs of the model, comparing it against global data sets of things like soil respiration and microbial biomass. And then this final stage is all about testing your new process within the context of an Earth system model. And I won't talk about this in a lot of detail because I'm short on time and because this is what you all do. This is what you live and breathe. But I encourage you to think about how to communicate what you do with an empiricist who's thinking about gathering data that they want to be valuable to a model. How do you communicate each of these different steps of connecting a simple model into the broader framework of an Earth system model, scaling that process up globally, and comparing Earth system model formulations with or without your new process? How do you express, how you're doing that work to an ecologist in a way that will help them to gather data that'll be useful to you? So with that, I hope this is a useful tool. I'm really happy to be talking to this group about this because I think, you folks do this all the time. You do this work all the time and you have these kinds of interactions. And I'm curious to hear whether this flow chart seems like a useful tool, whether it represents your understanding of how this process works, or whether there are glaring absences. But I hope in general that with tools like this, we can facilitate better bridging and better conversations between modeling and empirical communities, and ultimately incorporate more ecology into our system models. And with that, I'll take questions.
Thanks. We have time for a couple of questions if folks want to raise their hand now. There was a lot of discussion in the chat about the inclusion of fauna in models. Some folks mentioned that there's livestock on managed lands in some cases, or trying to get mountain pine beetle in. So that was what was happening in the chat. It looks like David has a question.
Yeah, if we could switch screen sharing over to Allison too, that'd be helpful.
This is, I really enjoy this, Emily. It's a great presentation. I'm going to use this framework when I have these conversations, which I have all the time. I'll just make one point. You said you have to ask whether or not it has a feedback onto climate. I'm not sure we're actually that strict about that. It's not necessarily 100% true that we don't want to include things that don't have a feedback on climate. If there's a use case where it'd be more beneficial to have it in the coupled system and getting the data out straight from the coupled system in terms of understanding impacts, those things can be considered too. And I'm thinking, for example, like heat stress indices in the urban model. No feedback on climate, but super useful to have embedded within the urban model to understand impacts on humans.
So how might you reframe that question in that conversation in order to, like, if that's not the threshold for what gets included, how do you define what the threshold for inclusion?
I'm not sure I can do it right now in a really brief, but I think where there's benefit to having the information integrated within the Earth system model simulation. I didn't say that very efficiently, but
yeah. Maybe more of it's on the impact side too, like understanding the impact. The heat stress is a good one. You want it in there for understanding impacts.
Any last questions for Emily?
Well, I think we're at the 15-minute mark. Feel free to use the chat, or we can cover more questions at the end. I'll let Allison Post start the next presentation. Take it away, Allison.
Great, thank you. Can everyone hear me? Okay.
Yeah, you sound great.
Great, thank you. All right, hi, I'm Allison Post. Thanks for having me here today. I'm a postdoc at Northern Arizona University with Andrew Richardson, and today I'll be talking about how data from the Phenocamp network can be used to evaluate and improve the community land model. So my talk, real quick, will be divided into 3 different sections. First, I'll introduce the Phenocamp network, what it is and how it works. And then I'm going to give a couple examples of how Phenocamp data can be used as a real-world comparison with CLM site-level output. And then finally, I'll give an example of how Phenocamp data can be used in model development. So let's start with the basic question. What is phenology? So the American Heritage Dictionary defines it as the scientific study of periodic biological phenomena, such as flowering, breeding, and migration in relation to climatic conditions. You guys see this every day. So phenology is, when the trees bloom in the spring, when fruits and vegetables mature, insect metamorphoses from caterpillars to butterflies, or animal migration, such as geese flying south for the winter. However, climate change is affecting the timing and pattern of many of these phenological events. So for example, due to warming temperatures, spring phenology events such as leaf out and flowering are occurring earlier in the season, and autumn phenological events such as leaf coloration and senescence are occurring later in the season. This is resulting in an overall lengthened growing cycle with cascading effects on nutrient and carbon cycles and the water cycle. And the study of phenology allows us to track these changes. In fact, the IPCC has said that phenology is perhaps the simplest process in which to track changes in the ecology of species in response to climate change. So, in order to better track these changes, our research group established a network of digital cameras to automatically monitor plant phenology. That is the Phenocamp network. So what we do is we and many collaborators have set up digital cameras that take images of many diverse types of ecosystems every 30 minutes during daylight hours. And some of these cameras also take images in the IR spectrum. What this does is it offers a direct link between what's happening on the ground, so at the scale of individual trees, to what satellites are seeing at a much coarser scale. There are currently over 500 cameras, with most of them in North America, specifically the continental US, but there's also some in Hawaii, Puerto Rico, Panama, and Europe. And they're across diverse ecosystems. And the imagery and data from these cameras is all available publicly here at this website. And some of these data sets have pretty long-term data now. For example, one of the first cameras installed at Harvard Forest in 2008 has now over 163,000 images. Here's an example of the types of images you get from the Phenocamp network. You get deciduous forest, evergreen forest, grassland, shrublands, and even some croplands. And new sites are being continuously added. Here's just an example of one Phenocamp site through the different seasons. This is a forest in Quebec. So you got spring, summer, autumn, winter. It's pretty striking to see those differences. But Phenocamp imagery is more than just those pretty pictures. We can also extract quantifiable values from the pictures. So digital images are stored as pixels that each have a numerical value of red, green, and blue intensity, which determines its visible color. So if we take a Phenocamp image and divide it into its 3 color channels, red, green, blue, we can determine greenness for a portion of the image, get an estimate of canopy greenness. So what we do is we calculate the relative brightness of the green channel against the overall brightness of the image. So green over red plus blue plus green, and we get a measure of greenness, or the green chromatic coordinate, GCC, as we call it. And we can get data sets like this. So this shows 7 years of canopy greenness at that site in Canada. So this is greenness, here's our years. This is in the winter when it's brown, there's not much green leaves. Here's spring green up, and then it slowly gets less green through the summer. And then here's fall color change and senescence. There are other couple advantages to Phenocamp imagery. For example, we can choose to just analyze portions of an image. So here, for example, in this shrubland, we're only looking at the shrubs and not the influence of the bare soil, which is a common challenge for remote sensing products. Likewise, you could also choose to only analyze certain functional groups. So here, we're just looking at the pine trees in the back and not the shrubs in the foreground. You also run into a lot fewer issues with cloudy days, since you're taking pictures closer to the ground. So then if we return to this greenness trend that I showed you, we can determine approximate dates of when spring green up occurs each year. So again, this is greenness, this is year. So using the amplitude of this greenness curve, we can determine the day of the year when either 10, 25, or 50% of maximum greenness has occurred. We can use a similar method to extract fall senescence dates. And this is important because the date of spring onset affects many ecological processes, and numerous models have been developed to predict it based on various climate variables, such as CLM. So now that you know how Phenocamp works, I will now compare Phenocamp data with output from a couple site level CLM runs at a couple neon sites. All right, so first we have Harvard Forest. This is a deciduous broadleaf forest in Massachusetts. This graph here shows GCC, shows the greenness over 3 different years. And here in the green is leaf area index, LAI, as predicted by CLM. As you can see, the patterns are pretty similar to each other. There's a couple years where it's predicted a little later than the actual greenness trend by about 10 days or so, but overall it's a pretty good fit. And we can also look at GPP from CLM, shown here in blue, and it also lines up pretty well with GCC, so that's good news. Now we get into the grassland ecosystems. So this is Kanza Prairie in Kansas. It's a tall grass prairie. And again, GCC is in black, and LAI from CLM is here shown in green. Not quite as good of a match. So let's break this down. Let's first look at the green up times for each year. We see that some years it matches up pretty well, but other years like this year, it's over a month early predicted. And then there's even greater disagreement for the overall seasonal pattern in length. So for example, in 2018, we get this big dip in LAI, which is not observed in the greenness. And then we see greenness extend way into the winter months. You know, this is January, which as we can see from the pictures, there's no green plant material at that time. If we look at GPP, it does agree better, which is good, but we do still see some issues. We still see that dip in 2018, and we still see GPP occurring during those winter months. So obviously there's a little need for improvement for modeling these grassland sites. So then that transitions me into how Phenocamp data can be used to develop new models. So what I'm currently doing is I'm using Phenocamp data to develop models that better predict that spring onset date in grassland ecosystems. This is important because grasslands account for 30% of North America and are large carbon sinks. And as we see in this map of the western half of the US, there are many diverse grassland types, from the Great Plains to the annual California grasslands to desert grasslands. And we need to be able to accurately predict timing of green up across all of them. So what I did is I extracted the 50% green up dates for all North American Phenocamp grassland sites, which are shown here. That's a total of 43 sites, and 195 site years, so individual data points. Then I worked on creating new spring models using the PhenoR package, which is created by a former postdoc of Andrew Richardson's, Kuhn Hufkins. And this package uses generalized simulated annealing to optimize parameters, and then uses AIC to compare models and determine the best fit model. So first, I fit my Grassland Phenocamp data to a suite of existing models. For example, the thermal time model says, oops, I don't want to see that yet. The thermal time model says that spring starts once a site has accumulated enough warm temperatures or growing degree days. However, as you can see, this model does not fit very well for the grassland ecosystems. And so let me just orient you here. This is the observed spring transition date. This is the predicted spring transition date by the model. This is the one-to-one line. This is the regression line. And then the colors here show which Köppen-Geiger climate classification each site falls in. So either arid, temperate, Mediterranean, or continental.
You have
Overall,
about 11 minutes, Allison.
Great, thank you.
Yep.
Overall, the root mean squared error is 35.4 days. And so there's obviously a need for improvement on just this simple model. So because grasslands are water limited, they often require precipitation inputs to green up in the spring. And so I created several new models that include precipitation. And this is my new best fit model. This one just uses temperature and precipitation as inputs, but that's enough to cut the root mean squared error in half to about 16.5 days. And importantly, it fits well for all grassland types with a single set of parameters, even these early transitioning Mediterranean grassland sites and these late transitioning arid grassland sites, about a 250-day spread in a green update. So to conclude, in this presentation, I showed you how Phenocamp data can be used to both evaluate existing models and develop new ones. By comparing Phenocamp data with the CLM site level output, we observed that CLM is pretty good at doing predicting dates for deciduous forests, but could be improved for grasslands. And Phenocamp data can be used to develop new phenology models, such as the spring phenology model that I am working on. So we hope this presentation introduced you to the unique value and potential of the Phenocamp data set, and hope you might consider using it in your own work. So here's the data or the website for the Phenocamp data again. Here's my contact info, and thank you. I'll take any questions.
Great, thanks. If anybody has a question for Allison, please raise your hand. In the chat, we'll mention that your figure is showing the GCC index versus what CLM predicted was his heart attack figure for the day, but it seemed like the GPP compared much more favorably. So what is the disconnect in CLM that allows that GPP to look okay, even while the leaf area looks so terrible?
That's a really good question. And to be completely honest, I am new to the CLM modeling world. So I'm hoping other people might be able to contribute to that. It's the first time I've looked at that. But from my understanding, LAI contributes to GPP, right? But then there's other variables that are also incorporated to get that value. Correct me if I'm wrong.
I can jump in a little bit. We haven't really had a ton of time to look at. So this is all like brand new stuff that's coming off the Neon project that we'll hear more about today. And my guess is that it's water stress that's causing that decline in GPP. So even though the plants have leaves, they're not able to, well, it could be water stress or they're buried by snow. Although I think you were looking at ELAI, not TLAI. Anyway, it's all pretty new stuff, but that double peak in grassland LAI is something that we've seen before, and we're seeing again at some of these neon sites, as well as sites that just aren't growing, like the phenological triggers aren't hit. So having the Phenocamp data in addition to the neon fluxes is really helpful to start diagnosing what some of these problems are.
Thanks, Will.
Montes here, would you like to start getting your slides ready as we ask the last question?
Yeah, I would say we have time for about one more question, so we'll go to Nancy Kiang.
Hi. Oops. Hi, thanks. Thanks. Been great talk. So I have a question about the data around the Phenocamp. Is there any irregular LAI on the ground measured at the same time? Because you've got simulated LAI and you get the timing with the Phenocamp, but how about the actual LAI?
The Phenocamp network doesn't take any other on the ground measurements. Although a lot of the NEON sites are associated with the Phenocamp network, and I know they take a lot more measurements. So that's something to consider.
Okay, thank you.
Yeah, great question.
Thank you.
Yeah.
Thanks, Allison. So we're ready for our next presenter, Montasir Maroof. Montasir, are you there? Can you share your slides?
Yeah, I'm here.
Great. We can see your screen. We can hear you. Take it away.
Can I start now?
Oh yeah, go for it. Thank you very much.
Good afternoon, everyone. I am Montasir Maroof, second-year PhD student at Auburn University, and my advisor is Dr. Sanjeev Kumar. So today I would like to give a presentation on a topic which titled is a NEON testbed for high-resolution climate assessment. So here is the outline for today's presentation. I will talk about the problem statement. I'll briefly discuss about model description and procedure. We'll talk about results, and at the end, summarize the findings. So the motivation of this study is to improve the water cycle prediction. And to improve the water cycle prediction, we need the high resolution model. So there's a hypothesis that high resolution topography, land use, land management, and boundary condition improve the land model output. So in this study, we evaluate the hypothesis. So to evaluate the hypothesis, we evaluated our model results against 3 observed dataset, which are Mira2, Larry, Neon, and also we compared our dataset with CLM 1 degree run to show the difference between high resolution and low resolution. Here is the brief model description. So the NOIMP and CLM high resolution, we run the model at 12.5 kilometer for both NOIMP and CLM high resolution. CLM low resolution, it has 100 kilometer, and for the reference dataset, Mira is 50 kilometer, Lady 1 kilometer, and Neon is a point scale dataset. And in terms of analysis period, NOIMP and CLM high resolution is 1980 to 2018. CLM low resolution is 1980 to 2014, and Mira 1980 to 2014, Larry is 2000 to 2014. And due to data unavailability, we choose 2018 to 2020 for Neon dataset. We used NL dust forcing for both NOIMP and CLM high resolution, and for CLM low resolution, GSWP3 was used. So to evaluate our results with the observed dataset, we calculated the ET, JJ bias and normalized root mean square error and compared these with Mira2. We also compared the bias and normalized root mean square error with Larry. We also did the climatology comparison with Neon, and we compared the residence time with CLM 1 degree run to show the difference between the residence time in high resolution and the low resolution. So here is the comparison of ET between high resolution and low resolution with Larry. So in the picture, it's very clear that CLF 1 degree couldn't capture the Southeast and the California regions. If we see the California side in the high resolution model, NOAA-MP and CLM high resolution, we can see there's a big difference in the South California, also some parts of the North, then high resolution and low resolution. The late ET shows the higher ET, but the CLM 1 degree couldn't capture this higher ET in that region. Whereas the CLM high resolution and NOAA-MP is close to the late one. If we also see the south side of the region, you can see the late ET get somewhere in between 135 to 180 millimeter per month, whereas the CLM 1 degree shows the ET is 90 to somewhere in between 105. But CLM high resolution and NOAA-MP performs really well in that region. So we calculated the ETJJ bias and compared this to the mirror dataset. And for the uniformity, we choose 1980 to 2040, this period, to compare our dataset with mirror. And we can see in CLM 5 degree resolution, the Southeast and the Midwest get really low bias than the CLM low resolution. Also, on the south side of California, the three models gets a little bit higher bias than a higher bias. And but CLM 5 and CLM high resolution gets really low one in south side Arizona and the southeast one. Also, the northern plains performs really well in CLM high resolution model. We also compared the bias with the Lady dataset. In the Lady dataset, the southeast get higher biases in the CLM low resolution. And here in the Lady dataset, we can see that Great Basin has some issues. In all three models, Great Basin gets higher bias than the other regions. But CLM high resolution performs really well in the southeast region and the Arizona one. We calculate the normalized root mean square error and compare this to Mira one. Here we can see the southeast and the northern plains has less normalized root mean square error, and Great Basin also shows relatively lower normalized root mean square error in high resolution model than the CLM 5 for low resolution. And this is the normalized root mean square error compared to the Lady one. Here we can see that the southeast side NOAA-MP performs really well in the southeast side, whereas CLM 5 is a little bit got higher values in the southeast side. So this is the summary from the normalized root mean square error. We can see that CLM high resolution get relatively lower normalized root mean square error when you compare our dataset with Mira 2. So it gets 0.178, whereas the CLM low resolution gets 0.193. And when you compare our dataset with Larry, the NOAA-MP gets the lowest normalized root mean square error, which is 0.226, and CLM high resolution is a little bit close to the NOAA-MP, which is 0.235. Again, CLM high resolution gets higher normalized root mean square error when you compare our dataset with Larry than the NOAA-MP and CLM high resolution. So from all the above results and images, it's quite clear that CLM5 high resolution performs well in most of the regions. And we compared the climatology mean with the neon ones. Here we can see that region 1, which is Harvard Forest, region 8, 6, 12, and 16 performs really well and is close to the neon observed dataset. But some of the regions shows really poor results. The 11.1, 11, New Wateries, and also Guan, it has some issues, and also it might be the reason because neon dataset is not very regular, so we did not get the all months dataset from the neon, so it could not capture the real scenario here. But still, it shows some issues, some of the regions, and performs well some of the regions.
You're at 11 minutes, Montasir.
So the soil moisture, we have calculated the soil moisture residence time and compared these to the low resolution to show the difference. Generally, with the higher resolution, the residence time increases. And this picture shows the fact here. We can see that CLM high resolution and NOAA-MP gets higher residence time in the California side and the Arizona side than the lower CLM low resolution. So in the California side and California, Arizona, and Colorado, we can see that CLM high resolution residence time is above 240. Also in NOAA-MP, it's also in between 208 to 240 days, whereas CLM high resolution, the California side, Arizona, and Colorado gets really lower residence time. And finally, if we summarize the results, we can say that CLM high resolution provides better output compared to NOAA-MP and CLM low resolution run. But the Southern California and Great Basin, these regions needs attention and further investigation because it gets higher bias and normalized root mean square error. Also, the residence time has increased in high resolution model than the CLM lower resolution model. So we are very thankful to USDA. This project is funded by USDA. And also we are very grateful to the NCAR team. Without their continuous support, we cannot develop this model. So thank you to all of them. And with this, I want to wrap up my presentation. If anyone has any questions or comments.
Thanks so much, Montasir. There were a few questions in the chat. There's been some active conversation. One was about why different forcings were used for the high and low resolution. Alan, do you want to follow up with that? Or were you sort of satisfied with the discussion in the chat?
Well, I don't understand why you use different data to force the high and low resolution model. You had high resolution data, so why don't you just downscale it and average it and use it to force the low resolution model? Then you'd be forcing it with the same data. Otherwise, you're comparing two different forcings to compare the model results.
We're trying to develop the lower resolution model. We want to run the lower resolution model with the same forcing data, but we got some issues, so we are working on it.
Okay, thanks.
Right, great. There's been some questions about the basis for the Leary data set and how we could better leverage some of these data sets for both development and benchmarking. I don't know if Abby, you want to follow up at all?
Oh, yeah, thanks. I was just asking some questions about what goes into the Leary data set, since all of these ET data sets are some kind of model as well, since evapotranspiration isn't directly measured. And so it's just good to have that in my head when I'm thinking about using it as a benchmark for how well the model is doing. Not that that means the model is doing well. And then Will asked or gave some information about if we should use this in ILAM. I was wondering if you know how similar the Leary estimates are to the MODIS estimates, which is, forget exactly what number the sort of MODIS estimates are. But anyway, there's all these evapotranspiration estimates, which are all kind of solutions to the surface energy budget that use some thermal imagery, remote sensing. Do you know how much Leary might differ from other estimates?
So you want to know about the difference between the Leary and the other models, the CLM high and NOIMP?
No, I'm actually asking about something you haven't shown. So very possibly you haven't looked into it, but do you know how, so, if you're considering Leary as the benchmark here that you're comparing high resolution or low resolution versions to, how does Leary compare to other observationally based estimates? Since each of those are sort of in themselves a type of model.
So sorry, can I get your questions again?
Maybe in the interest of time, we can continue with that in the chat. And as you get your screen taken off and Adriana gets hers on, maybe you can answer Dave's question about why would residence time be dependent on resolution? Could this possibly be due to forcing differences?
We're
Should I
doing,
go, or-
we're doing
Yeah.
pretty good on time, but
Yeah, I think
maybe it's a good time to transition, and those three can take it, have a conversation, take it offline, as they say.
likewise. Great. Thank you, Montesir.
Yeah, thank you, Montesir. And Adriana, you're next. Take it away.
Great. Can you guys see the presentation version of my
Yes.
PowerPoint?
Yep.
Great. Yeah, so I'm Adriana Foster. I'm a newish project scientist at NCAR working with FATES. And so I've been investigating FATES at some of the NEON sites. And so I'd like to walk through some of those methods and then some pretty preliminary results from the work. So, with a model like FATES, we are necessarily in the business of capturing ecological processes across multiple scales, such as cell level processes of photosynthesis, leaf level processes like stomatal conductance and impact on boundary layer dynamics. We also have tree and stand level processes like allocation and competition. And then at the landscape scale, we may want to model emergent processes like these fur waves as a result of all of these multi-scale processes. And the NEON sampling design provides a way to benchmark FATES at these multiple scales. So for fast processes like photosynthesis and transpiration, we can use the NEON eddy covariance tower data. For allometry and vegetation composition, more related to forest dynamics processes, we can use the NEON inventory data or aerial data. They also collect information on below ground conditions, as well as a ton of other data, which you can check out on their data portal. And so I'm initially applying FATES at a handful of these NEON tower sites. And today I'm going to show results for Bartlett Forest in New Hampshire, Harvard Forest in Massachusetts, and then mainly Bonanza Creek in Alaska. So one of the difficulties with getting FATES up and running in a new location is choosing PFTs and parameter sets. And we can try to leverage existing databases to do some parameter calibration. So in terms of the PFTs that I wanted to use, I could have just said every species is its own PFT. So if I did that for only these 3 sites, that would be about 26, which is quite a lot to parameterize and to model. So here you can see observed basal area at the 3 sites colored by species. And so I initially did was to combine species by genus and form, as well as shade tolerance. And so I came up with 8 PFTs across these sites. So not all of the sites had all 8 PFTs present at them, but they shared quite a few.
And so I initially wanted to calibrate FATE's allometry parameters, including those for height, above ground biomass, leaf biomass, and sapwood biomass. And to do that, I used the Biomass and Allometry Database, or BAD, which is a large database of plant allometry characteristics. So on the bottom left, you can see a map of locations that have measurements of my target allometry characteristics. And then I also filtered those to include only temperate and boreal tree species for this study. And now on the right is just a pairwise graph of the different allometry characteristics. And in general, there's quite a bit of height measurements, less woody and leaf biomass, and really not a lot of sapwood biomass measurements. So for this calibration, I used Bayesian Markov chain Monte Carlo statistical methods to calibrate the parameter values. So essentially what this does, it samples many thousands and thousands of parameter values to approximate a posterior distribution of your parameters. So this type of calibration is really nice because we can not only get at sort of our answer, our mean estimate, as well as the uncertainty using these distributions. So here we can see the median plus the 95% confidence interval in red for estimated parameters for height allometry. For this, I used the Martinez-Cano 3-parameter allometry. So we can see the separate parameter estimates for a shade intolerant broadleaf deciduous tree on the left versus a needleleaf evergreen shade tolerant tree on the right. Okay, so let's look at some runs with FATES at these sites where we can run the model from bare ground initiation. So here are results from the 3 sites just using default FATES parameters. So this is plotting basal area on the left over time compared to the observed basal area in these bar charts on the right, all colored by PFT. So for the site in Alaska, we're getting total basal area, it's kind of about right, but we're missing birch and aspen, perhaps due to issues with not representing topographic variability, as this is very important in Alaska. At Bartlett Forest in New Hampshire, we have an outsized amount of spruce and pine in the blues, and we're missing this maple beech PFT, which is our shade tolerant deciduous PFT. And then for Harvard Forest, it looks okay, though again, we're missing some deciduous species. So in general, we get a very quick succession from broadleaf deciduous to needleleaf evergreen species with these default parameters. So here we have basal area output from bare ground at Bonanza Creek using the mean value of the calibrated parameters. So we're still missing this birch and aspen PFT, but I think the basal area actually looks better for the spruce PFT. In Alaska, we would actually expect kind of low basal area for spruce at this site, especially since it's black spruce. Here we can look at observed size structure on the left and then FATES output on the right for the same site. And this is at year 2018, so 100-year stand age. So we can say that, you know, we do have a ton more small trees in the model than we do in the observations. If we take away that 0 to 5 centimeter bin, we see that we don't actually have as many larger diameter trees in the model as we do in the observation. So something to work on going forward. And so in many cases, we may actually want to initialize FATES to current conditions. So we heard a lot of talks yesterday about internal variability. And one source of major uncertainty and variability is initial conditions. So NEON takes inventory measurements at several plots at each site. Here is a plot of locations of trees on one plot at Bartlett Forest with the size of the dot corresponding to the tree diameter, the color corresponding to its PFT. So we can take this data and easily convert it into a structure that FATES can read in as initial conditions for patches and sites. Okay, but you know, what if we wanted to initialize more than just at the handful of inventory plot locations? So for that, we could leverage the aerial data that NEON collects over these sites. So for instance, NEON collects aerial LiDAR data at its sites. And from that, you can get these cool 3D point clouds of forest structure like this here, which is from the Lower Tea Kettle site in California. And again, we can use this data to initialize FATES across broader scales with a bit more post-processing. So here is a canopy height model, which is just a map of tree height generated from the LiDAR point cloud data. And this is for Bonanza Creek in Alaska. So we can see the Flux tower, this red dot, and then the squares are the inventory plots surrounding the tower. So if we zoom into this canopy height model, I used a crown delineation algorithm to detect individual trees within this canopy height model. So this method uses a moving window to find points of local maxima within the canopy height model to locate individual trees. So with this method, we can get a distribution of tree heights surrounding the neon tower. Okay, great. So it's not too hard to pick out the trees from this point cloud and use methods to determine their height and the distribution of heights. But just looking at this point cloud alone, we don't really know what PFTs they are. So for determining PFTs, we can try to leverage the hyperspectral data that NEON also collects over its sites. So here we can see an RGB hyperspectral image over Bonanza Creek. So to do the actual classification, I trained a random forest model on the common vegetation indices calculated from the hyperspectral data using known PFTs and locations of trees that NEON tags as part of their inventory process. And so at a site like Bonanza Creek, where we're just looking to distinguish between spruce versus birch/aspen, which is essentially just deciduous versus evergreen. So here on the left, you can see a graph of reflectance spectra across all the wavelengths for spruce in blue and aspen in yellow. And so there's, you can clearly see divergence. So the random forest does a pretty good job here with 92% accuracy against independent testing data.
You're at about 11 minutes, Adriana.
Oh no, okay. In contrast, at Harvard Forest, the PFTs are a lot more similar, so it's harder. So we'll have to work on that. Back to Alaska. So here's our canopy height model and our identified trees. And I'm actually pretty happy with this because if you look at this photo, you can see sparse spruce trees here, which are the blue dots. And then you can see this dense birch aspen in the background, which I think corresponds to what we actually classified it as. So here is the GPP tower observations from initialized runs with the face observations in blue and the neon tower observations in red. So it looks pretty good. And this is actually using default Fates parameters. So I'm going to quickly run through parameter calibration and uncertainty. So I had these posterior distributions of parameter values for several allometry equations. And then I pulled from these posterior distributions to run several runs of Fates pulling from these distributions. You can notice there are correlations, which is okay because we're using the joint distribution, not individual marginal distributions of each parameter. And so here is GPP of our ensemble in black with the median plus confidence interval. The Fates out of the box default parameters is in blue and the neon observations are in red. So we're actually overshooting GPP now. This is with the sapwood allometry. I was a little worried about sapwood because it had so few observations. So here it is without the sapwood allometry. Now we're undershooting, so definitely something to look into. Maybe obviously this is looking at the parameter values on the x-axis versus the annual GPP. They're most correlated with, GPP is most correlated with leaf and sapwood related parameters, as opposed to not very correlated with woody parameters over just 3 years of GPP, though I would expect it would be more influential over longer periods of time. And so in the future, this is just some initial steps I'm taking at these neon sites, but I have plenty to investigate with regards to parameter uncertainty and successional dynamics. I want to run these sensitivity tests with bare ground setup to see how they impact longer term dynamics. And then I'm also hoping to investigate how to apply these Bayesian methods to photosynthesis parameters and maybe with Fates hydro and nutrients. And then there are plenty of other forested neon sites to do this workflow at. So thank you.
Great. Thanks so much, Adriana. We have one question in the chat from Ensheng. For lacking of large trees, it might be a result of the allometry. For some formulation, as a tree gets larger, its diameter growth decreases because of respiration costs and allocation to leaves and fine roots, depending on the allometry parameters. I guess it's more of a question. I don't know if you have a response or follow up to that, Adriana.
Yes. I agree. I think. Can you read that again? Sorry.
Sorry, it's just in the chat. So
okay, but I think I agree that the diameter theoretically should slow. We know, well, in temperate forests, we see that tree diameter slows as at diameter increment slows as the tree ages. Yeah.
All right, great. Dave looks like he has a question.
Yeah, so nice talk, Adriana. So I wonder, how concerned should we be if we are not getting the size, can't produce, the default model can't produce the size distributions realistically, can't get the big enough trees, but yet then you're going ahead and initializing those large trees. Should we be concerned about what comes out of the model once you initialize those trees with realistic?
Yeah, I mean, I think I definitely want to investigate more as to, why we're getting GPP, we're like pretty spot on with the default parameters, but then we're not getting necessarily like the species composition and size class distribution that we see. And the other point to note is that we are only looking here at like 20 plots, and that's like a pretty fine snapshot of forest dynamics. And so it could be that like looking at this basal area plot for Bonanza Creek, like that forest definitely exists in Alaska. So I don't know. Yeah, and it doesn't really answer your question, but it's definitely something to investigate. I do think initializing to current conditions is a way to kind of benchmark different parts of the model. Rosie's method that she's been working on with all these different fates modes is kind of akin to that. And so I think, we can kind of hold certain parts constant while we work on, say, photosynthesis. And then if we're looking at more of these slow processes that would build up the, correct stand structure, that might mean that we have to hold other things constant. Yeah.
Thanks, Adriana. We probably have time for one more question. Ling Cheng wrote in the chat, as you use the MCMC, how does the computational efficiency as fates need long times to reach stable state for each run? And how is the PFT's coexistence captured during your calibration?
I'm not sure I understand. So the MCMC, yeah.
So first, thanks for your talk. So I see you use MC to calibration, based on my understanding that for each parameter calibration run, the phase needs maybe 200 or 300 years to reach stable state. So
yeah, so
it's long time. So I'm wondering how the efficiency use MCMC if you try 2,000 combinations of parameters.
Yeah, so I should have clarified. I'm actually doing this MCMC on just the equations for the allometry. So just height. So I'm doing them kind of independently. So I'm not actually, this was not feeding it into the model. I'm doing this sort of outside, independent of fates itself. So in theory, this should, if we get this improved, it should improve fates. Kind of the idea was that we get a posterior from this, which becomes our priors for fates and moving into fates. But yeah, because it does, running the 3 years doesn't take that long. I can do that very quickly, but running a whole bare ground to 100 years, that could take a few hours for a site. So that's definitely an issue. But for this, it's nice because I actually have data that I can just look at this one equation by itself. Yeah.
Okay, thank you. My another question is when
I think we probably I think we probably have to transition to the next talk. But thanks, and it's been great to see the conversation continuing in the chat. And as Elizabeth noted, you can save the chat by pushing the 3 dots, or you can contact her after the meeting to get an archive of the chat, because there's been a lot of good discussion there. So, all right, Donica, do you want to go ahead and take over? I don't know if it's you or Nagin who will be sharing your slides, but
yeah, we're gonna take turns. So let me have to share my screen. I will share this. That looks like, okay, so here we go. Yeah, so I'm Donica Lombardazzi, and I am a project scientist at NCAR in the terrestrial sciences section. For those of you who don't know me, I know many of you do. But today we're going to be talking about a big collaborative project between NCAR and NEON, and you've already heard several examples just in the last hour or so. You've heard several examples of how people are already using this tool that we've created. And so we want to give you a little bit more of an overview. And I've asked Megan, Dave Durden, Will, and Gordon to join me in this presentation because we have been working hard at developing this new tool in collaboration with several other people, including additional data people at NEON and software engineers here at NCAR. So I'm going to hand it over to Dave for just I guess we'll start by giving you just a quick overview of what the tool is, and then we'll dig in. So I'll hand it over to Dave to explain a little bit about the NEON data and data processing.
Yeah, thanks, Danica. So when we started this project, we knew that we had to gather all the inputs necessary for CLM from the NEON data store. And that started with the meteorological inputs and then gathering the site characterization data, vegetation traits, and then also collating data for model evaluation. I'd like to point out that principally right now, the data consists of the 2021 NEON data release, but the 2022 NEON data release just came out last week, and I'm currently in the process of updating to the new data release for our version 2 data set that should be coming soon. And additionally, I developed a gap filling workflow for the meteorological data and then partitioning workflow for the model evaluation data, such as the fluxes coming off the tower. Next slide, please. And so this dives a little bit more into the workflow. So first we start by gathering the data from the NEON API using open source tools that we've developed in R and Dockerized that are all available via GitHub. And then we perform kind of a redundant data stream gap filling, utilizing the wealth of data that we have at the NEON sites. And then whatever can't be gap filled with the redundant data streams are then gap filled using other traditional approaches for gap filling. And then data are converted to NetCDF and standardized units, and then output for CLM ingestion. Additionally, we've also been partnering with some of the with people working on ILAM through Rubisco, and then the Phenocamp project, and recently the Ecological Forecasting Initiative is looking at ingesting some of our data files into their challenge for this year. So next slide, please. Lastly, I want to talk a little bit about where you can find additional information about the project and some of the data availability. So the first bullet point here, you can see we have a NCAR NEON community collaborations page where you can find out more about this project and kind of explore some of the tools that will be talked about later. Additionally, we have example prototype data sets available now via the NEON prototype data portal. Right now, those are very preliminary. We only have one site. But if you're interested in a more substantial data set, all the data are available via an S3 bucket. And at the bottom here, you can see the URL for the listing of all the available data. Okay, thanks. I'll pass it to Danica.
Yeah, so we have been ingesting the process NEON data. And so, as Dave said, that includes meteorology, site characteristics, and flux tower data for model evaluation into a containerized model system. And in this containerized model system, we can run CLM simulations to get ecosystem states and fluxes. And so these data can, both be evaluated against NEON data and can also be used to supplement NEON data for observation or for ecosystem processes that are not being measured through the observational network. So that's really exciting. We also provide tools for analysis within this new tool. And so these come in the form of self-guided tutorials. It allows you to download the Neon data through this tool. So if you want an easy way to download Neon data, or another easy way to download Neon data, you can also work that through the tools for analysis. We walk through how to create plots, and you can evaluate the differences between Neon tower data and CLM simulations. And we'll be walking you through that in just a minute. So I just want to highlight here that what we're using for the containerized system is CESM Lab, which is the CESM Docker container. So you have to download Docker and I guess have an account. And then once you do that, the CTSM tower simulations or the CTSM simulations at Neon tower sites are available. So this is actually a screenshot of our tutorial. Beta version of this tool is already released and is available on a website that we can add to the chat in a few minutes. But yeah, this just walks you through how to run a site simulation at Neon, and it comes down to basically 1 or 2 lines of code. So it's really pretty simple these days. So what we'd like to do next is to have you help us select a site to look at some of these data for. And so we're going to be looking at latent and the energy balance, focusing on latent and sensible heat fluxes, and also we'll focus on net ecosystem exchange. And then we're going to look through each of those at one of these 5 sites that we've selected just to look at. And so Elizabeth is going to launch a poll for us so that we can first select if we want to look at the latent heat flux or net ecosystem exchange first. We will look at both. It looks like about almost 70% of people have responded, and the response rate is slowing down. So maybe we can, oh, there it goes again. Okay, yeah, maybe we can end the poll and show the results. All right, so we are going to start with net ecosystem exchange by looking at that. And next, if we can launch the next poll, we'll choose which site we're going to look at net ecosystem exchange for. And so I kept this map up because these are the 5 sites that we're going to let you choose from. So Elizabeth, if you can launch the second poll.
1 second.
Okay. Great. Right.
I don't know why, but I get to see everyone's stuff coming in, and I'm like rooting for certain sites.
Yeah, yeah, me too. We have about 70% of 75% participation so far. That's exciting. Ooh, 80% now. All right, if you haven't voted, get your votes in. All right, we have just about 90%. So why don't we share the results of the poll? And it looks like our winner is actually Kanza Prairie, which is surprising to me because it's a grassland site, but that's exciting that people want to look at a grassland site. Okay, so let's stop sharing the poll, and I will stop sharing my screen, I think. Wait, how do I stop? Oh, stop sharing. There we go. And so next we're gonna transition over to Megan. She will start sharing her screen next. And what we're going to do is look at these interactive plots that Megan has created. So Megan, we're gonna start with net ecosystem exchange. So we'll look at these interactive plots that Megan created. And I just wanna highlight that these will be available on the Neon tower site, or the Neon website, these simulations. And so you can look through these interactive plots in that way in the future if you would like, and we'll walk through some of the results now. So let's see. We are looking at Kanza, and I guess I wanted to start by highlighting a few trends and orienting you to this plot. So this is net ecosystem exchange, like I said, for Kanza Prairie. And you'll notice that the blue line that shows the data measured by NEON at the NEON tower site. And then the red line is the simulation by CLM. And so anything that is below the zero line is going into the land surface. So that is a net uptake of carbon flux, and anything that is above that zero line is going back into the atmosphere. So that's net flux back into the atmosphere. So I guess I wanted to start by asking Dave here. So Dave, what is NEON measuring when it's measuring NEE? And what do you think about the variability in observed NEE?
Yeah, so we're measuring the net ecosystem exchange, which is comprised of both the turbulent exchange at the site. So thinking about the eddy covariance system at the top of the tower, and the storage component, which is comprised of the profile of measurements going down into the canopy or above the surface. And so right now we've been kind of working on improving some of the data quality. As you can see, there are some potential outliers in the data set as well. And we're hoping that in this 2022 data release that some of these will be resolved, especially in the turbulent data set, but we're still working on refining the storage data set itself. But we felt that it was important to have both the storage and the turbulent component here to compare against the model.
Great, thanks. And so what I'm noticing here from some of these trends is it doesn't look like net ecosystem exchange has a strong drawdown in 2018 in the tower data, but it's stronger in CLM. But then for 2019 and 2020, the data look more similar. The simulated and observed data look more similar. And so I guess, Will, this next question is for you. What do you think about the modeled variability and what it is that the model is simulating to understand exchange?
I guess after seeing Allison's plots for Kanza, I'm impressed that we look as good as we do. You can especially look at, well, I guess a couple things. One is that we're Megan zoomed in here for 2018. I guess I'd have questions about the data quality in terms of what we're getting from NEON, but I think Dave has said in the past that the data quality is usually better kind of later in the time series. So maybe 2020 is a better year to zoom in on here, Megan. Any kind of carbon cycle variability in the model, especially in the net fluxes like this, is really reflecting the component of two larger fluxes, which I guess is an interesting distinction between NEON, which is really measuring NEE, and CLM, which is actually simulating GPP and ecosystem respiration, and then NEE is the difference. But it seems like, by and large, we're getting the low point of this drawdown, and then kind of after the peak greenness kind of goes back. But so the component flex that might be interesting to look at here would be GPP. I don't know if we have how much time we want to spend going through these.
Yeah, well, we do have GPP data, and I was going to ask Gordon if he could tell us a little bit more about how GPP is simulated in CLM, and why CLM may or may not match the observations.
Yeah, so what I would say is, particularly if we look at 2019, that's a pretty good simulation of GPP. Maybe it's a little bit overestimated, sort of in July and August or so, but it's looking pretty good. And so what we want to do is we want to understand, are you getting the right answer for the right reason? And in CLM, GPP is a combination of two separate sort of processes. One is leaf level photosynthesis itself, and then the second is the scaling of that leaf photosynthesis over the canopy. The leaf photosynthesis is the Farquhar for C3 plants, at least, is the Farquhar photosynthesis model coupled to the Medlin stomatoconductance model, and that's solved to give you the photosynthetic rates. So things that could be going on at the leaf level would be like a vapor pressure deficit, for example, as it affects stomatoconductance. It could also be soil moisture. Those type of things could be affecting the leaf photosynthesis rate. But then there's also the scaling of that photosynthesis over all the leaves in the canopy, which is related to the radiative transfer in the canopy, the distribution of nitrogen in the canopy, the actual nitrogen concentration of the canopy, and things like that. All those at the leaf level and the canopy scale can contribute to why the model is looking good or bad at a particular site and time of year.
Yeah, so that's really interesting. So CLM is using a process-based model and scaling up from leaf level photosynthesis models to canopy photosynthesis. But I'm pretty sure that the neon flux towers don't simulate or don't directly measure photosynthesis, right? And so how are we, Dave, how are we seeing this observed GPP from the Kanza Prairie site?
Yeah, so as part of the processing, we have to calculate and model GPP using the NEE that we derive from the turbulent and storage fluxes. And we're currently using the Reichstein et al. Nighttime partitioning model. But the different model that's used could potentially have different results output for GPP. So in the future, we hope to kind of explore what differences there may be using different partitioning models.
Great, thanks. So this is just really a high level overview of carbon fluxes that are modeled and measured. And I want to spend a little bit of time also being able to look at the energy fluxes and specifically, latent heat flux. So I'm wondering if we can launch a second poll. Do we want to use the same sites, or do we want to look at Kanza Prairie, or do we want to look at a different site and see what latent heat flux looks like for a different site? So if Elizabeth can launch a poll, and if you can help us select another site, that would be fantastic to all of you in the audience. And Aaliyah, I did see that you had a question about spin up while we're waiting for this poll to get launched. And we are spinning up the tower. We are spinning up the simulations by cycling over the flux tower observed meteorology. So that's how that is working.
While I'm doing that, I just wanted to point out that Bartlett Forest was a really close second, but I went ahead and that's open again now.
Yes, thank you. Right. We're getting close, 70% participation. If you haven't selected a site yet, please do. Okay, I think responses are slowing down, so why don't we end the poll for the time being? And Elizabeth, I don't know if people just got excited about Bartlett, or if your comment made people select that, but it does look like Bartlett is our tower of choice for the latent heat flux and the energy fluxes to get to understand that. So I guess if we can stop sharing that, then maybe we can look at the graphs from Negan. Okay, so Negan has moved us to the latent heat flux plots for the Bartlett site. And again, just to orient you to these plots, the red line is the CLM simulations and the blue line are the neon simulations. And so you can see the cycle of latent heat flux for this particular site. And so, yeah, Dave, one question that I didn't ask you on the net ecosystem exchange, but you kind of alluded to, are that neon data can be missing or sometimes seem to be outliers. And you don't always see that as well here, but there's one point that has negative GPP in 2019, where Negan is circling. And so that's one example of where there might be an outlier, for example. And so I'm wondering, can you tell us why this why we might have outliers or missing data from Neon?
Yeah, so during the last data release, we had some overflagging issues, especially in the Flux data products. And so in order to circumvent kind of just removing an excessive amount of data, I kind of did a light filtering on this data set. So there are some potential outliers within this data set. In the upcoming version 2 of this data set, we have refined our flagging algorithms, and we believe it'll do a much better job of kind of eliminating some of these outliers and allow us to use the final data quality flag provided through Neon to do some of the filtering. And as you can see, as Will talked about later, usually the data look better as we go forward in time with Neon, as we've improved kind of our dashboards and the ability to respond to the need for corrective maintenance in the field. But we were kind of slowed down somewhat by COVID. And so right in 2020, you can see that for a while we weren't able to access the site, and could result in the sensors going down and having a little bit slower response time on getting sensors back out to the field.
Thanks, Dave. That's really interesting information. So if Megan, if you can go back to the full-time series, one of the other things that I'm noticing is that it seems as though CLM is underestimating latent heat flux for these tower sites. And so Gordon, and that's, I mean, it's particularly evident in, for example, 2019 and in 2020. And Gordon, I guess I'm wondering if you can explain to us why there are differences between the simulations and the observations.
Yeah, so here the thing you do want to look at right away is the components of the latent heat flux. And in CLM, that consists of transpiration, evaporation of intercepted water, and soil evaporation. So one of the first things to do is to look at the three different parts of the total latent heat flux to see which one is the more dominant one and at this site. And so Megan, I think
can you switch to that?
Yeah, okay. Yeah, and so here what we see is actually something sort of fairly typical across sites with CLM, is that transpiration is in the sort of the lighter green is the dominant component of the total flux, and so that's really ground evaporation and canopy evaporation of intercepted water are much smaller terms, at least in this sort of summer growing season. So that's really pointing to transpiration at this site as a really big factor. And I think we can do something here. Megan, if you pulled up GPP, I think we would see that that is also very low biased at this site. And so we would actually see that this is, again, sort of very common in CLM. If we're underestimating latent heat flux, we also tend to be underestimating GPP. And that's is what you're seeing here. Particularly like in 2020, you're seeing, and a little bit in 2019 also. And that tends to be really common, low transpiration and low GPP, which sort of points to things like leaf area index, stomatic conductance, integration of leaf processes and stomatic conductance over the canopy as a potential factor in both of those.
Yeah, so I mean, that's a great point. And I guess I have a follow-up question for Will. If transpiration dominates, and part of that's connected to GPP, how can we tell if CLM is getting transpiration right?
Yeah, that's a hard one to try to figure out. But since Gordon kind of highlighted that transpiration is the dominant flux, and we're underestimating GPP, it makes me think that there's a good chance we're also underestimating LAI. And I think in the chat somebody was asking if there are LAI data connected to Phenocamp, and there's not LAI from Phenocamp or from Neon data products that are available right now. But I think, oh cool, you got this to plot up more clearly, Megan. So on the secondary y-axis here is exposed LAI, which it's maxing out around 4, which probably seems relevant to Bartlett. So if the LAI seems okay, the GPP is low, and the ET flux is low, it makes me think that something with stomatic conductance maybe isn't quite right. The plants should be moving more water than we're letting them right now. So it could be something related to stomatal conductance. And then,
hey folks, my timer went off to give you your time warning.
Awesome. Yeah, as you guys can kind of see, you can dig through these plots and kind of click through stuff forever. Like, it's so cool and it's so great, I think, to the way that Megan's put all this together. I'm super excited about it. I'll stop.
Yeah, well, so Will, I wanted to ask just a quick follow-up question, because then I also want to look at net radiation. But
yeah,
I guess one of the things that I wanted to highlight is that this site is a northern mixed hardwood, and we just represent this as one plant functional type.
Yep.
Could that contribute? I mean, the parameter values that we're using for some of these simulations, could that potentially contribute to some of these mismatches?
Yeah, totally. Like, we're trying to represent this is probably a boreal forest PFT or a temperate evergreen PFT. I'm not sure which one it would be.
Deciduous, but yeah.
Oh, is it deciduous? Yeah, and so we're trying to represent all of those species that are representative of that PFT at the site, and we could be mismatching it. So again, Adriana's work trying to parameterize, getting more appropriate parameterizations with fates would potentially be valuable to do with the big leaf model here.
Yeah, that's yeah, exactly. And so ultimately what goes into the model can also contribute to biases. And so there are things like net radiation, for example, that the model is simulating that we can check against neon data. And so, Gordon, I'm wondering, if you can tell us a little bit about net radiation and what other things might be contributing to these biases.
Yeah, so one of the first things you should always do when looking at the model output is look at net radiation, because that's particularly for latent and sensible heat flux, that's so fundamental. Net radiation is not input to the model, as Danica said, it's calculated from downwelling solar radiation and albedo, and then atmospheric long wave radiation, and then the surface temperature and the emitted radiation from the surface. So in some ways, looking at this model to observed at the Bartlett site is looking really pretty good, except that there is a low bias in the model with net radiation compared to the observations. You can see it particularly in that sort of July period, but you can also see it in the scatter plot that Megan has over on the side. The model tends to be, probably it looks like 10 to 20 or 20 to 30 or so, maybe watts per meter squared, low bias. And so that's then contributing to the low bias and latent heat flux. If you don't have enough net radiation, then you don't have enough energy available for latent and sensible heat flux. So that could factor in it. There's also then the partitioning of that net radiation into sensible and latent heat flux, through the Bowen ratio. And that then, again, depends on things like leaf area, soil moisture, stomatic conductance, variety of things like that. But this is actually not such a bad simulation of net radiation, but it does point out the trouble we have in comparing to observations again, because if you have a low bias in net radiation, then you're going to see biases in sensible and latent heat just because of that alone.
Yeah, so one of the things that you were just mentioning is the latent and sensible heat fluxes, and we're looking at net radiation, and that looks pretty, like we simulate that pretty well, maybe a little bit of bias, but there's larger biases in latent heat flux. So what do you think will happen to sensible heat flux?
Well, fortunately, it turns out the way we would hope it is, which is that, if latent heat flux, just from an energy balance point of view, if latent heat flux is underestimated, then sensible heat flux tends to be overestimated. And that's exactly what you're seeing, again, in this July 2019, July 2020 period. The model's high. Again, it's interesting, these biases we're talking about really aren't that high, to be honest with you. We're talking about biases of like 30, 40 watts per square meter. And I would actually, we haven't talked about this at all, but I would actually say that's well within sort of the measurement uncertainty, the measurement error itself of the instrument and the processing and things like that. So we are in some ways talking about real subtleties here of the model data comparison.
Yeah, I totally agree, Gordon. And I guess I just have one final question, because what you were just describing is that the latent and sensible heat flux balances I guess the energy cycle, energy budget needs to close within CLM. And so Dave, I'm wondering if this is true for the neon tower measurements.
No, actually, each of the measurements of the sensible heat flux and the latent heat flux are measured independently, and typically the energy balance doesn't close at eddy covariance tower sites, and so I think that's a great, and that's one of the big issues I think in comparing data versus model outputs. So and I think it's a great topic for further discussion and exploration.
Okay, well, I know that we're a little bit over time, so I'm going to wrap up there. And I would just like to thank everybody who has answered all of my questions about this. And I would like to emphasize to the audience that this is a tool that is available for use. We're still working on finalizing a bunch of things. And so please feel free to start using the tool. We can put the link in the chat. And just, I think that this really highlights how exciting it is to be able to run simulations at these neon flux tower sites, and just how challenging it can be to compare model simulations with observations. And just highlighting that even though, yes, models can be biased and there are issues with that, there are also some challenges with data as well. And so we have to keep both of those things in mind when we're thinking about and evaluating simulations. This is a great tool for being able to improve both the observations and the model simulations. So yeah, I see that we'll put the link in the chat. And so I'd just like to thank you all for your attention, and let us know if you have questions or start using the tool. Great, Megan.
Well, thanks, Danica, and thanks to all the speakers in this session. You all started 5 minutes late, so you were really right on time. Gretchen, how should we proceed? Was there anything in the chat that didn't really get rectified, or should we go to showing of hands and stuff like that?
I guess there was one question about whether there's plans to make a Fates version of this visualization tool.
Yes. Yeah, we are hoping to add Fates, and I know that Adriana and Megan have been working on some of those capabilities, and so we're really excited for that. I think it's going to be a step for a little bit down the line, but we're really excited about the capabilities to potentially use Fates at these neon flux tower sites as well.
Are there other questions? It seems like a lot of the chat has been sort of resolved. But I guess we also have only a few minutes left. So if there's other, broader questions across the speakers, that'd be welcome as well. Will?
I might just add a comment. So definitely, I think there's a strong desire to add Fates to this. And the one thing I'll say is this was a 1-year NSF investment to NCAR and Neon that we're pretty much done with. And so getting it continued is always kind of the next challenge. And so we're working on having those conversations, but getting support from the community, having people use this tool, talk to program officers, I think it's super exciting. But right now there's not funding to keep it going. So it needs energy, both from the community and then funding to kind of keep it going.
Thanks, Will. Other questions or comments? All right, well, I think this has been a great session. We can sort of end here for the day, and we'll regroup tomorrow at 9 Mountain Time for, it looks like, a discussion focused on sort of impacts and land use change, land management issues in CLM. Ryan, do you have anything you want to add, or Will, do you have anything you want to add before we close?
Not me. They're great. They're all really great talks this afternoon. So just thank you.
All right, great. Well, we look forward to seeing everybody tomorrow. Take care.
Thanks.
Thank you.